Our today's topic is working of AMI and CMI in human body. The immune system of our human body is made up of lymphoid organs, which are made up of lymphoid tissues, which are further made up of lymphoid cells. Lymphoid cells are of two types, the T cells and B cells. The T cells makes up our CMI while the B cells form our AMI. Both of these cells are formed in our bone marrow originally. The lymphoid organs are two types, primary lymphoid organs, that is bone marrow, that is main, and thymus gland. The secondary lymphoid organs are our spleen, our tonsils, Pierce patches, lymph nodes, appendix, MALT, that is mucosa associated lymphoid tissue that forms 50% of our lymphoid organs. The T cells and B cells, let's talk about them. So both of them originate in our bone marrow. The immature T cells goes to thymus gland for maturation and multiplication. Then the mature T cells that comes out of our thymus gland are of further four types, that is the T messenger, the T killer, T suppressor, and T helper. The T helper is very important. The CMI forms about 60 to 70% of immunity. The mature B cells comes out of our bone marrow, means the B cells mature in our bone marrow, not just formed. So B cells are of two types, the B messenger cells and B plasma cells. The B messenger cells form the memory of our pathogen that is attacking us, and the B plasma forms antibodies, which is the basis of our AMI, that is 10 to 20% of our total immunity. Let's rub this. So the next thing we will see is the antigen presenting cells. These are special kind of cells. Now these cells process our antigens and represent the antigens on its surface through some magic receptor, that is MHC receptors. Examples are macrophage, B lymphocyte, and dendritic cells or Langerhans cells. They are found on our skin. So as any pathogen comes, it interacts firstly with these APC cells. Suppose a pathogen is trying to enter our body, so first it will come in contact with the APC, which has two MHC receptor 1 and 2. MHC1 deals with endogenous pathogens, that is tumor or virus that is using cellular machinery, and an organ that is being rejected by our immune system simply. So while the MSC2 deals with exogenous pathogens, there are many bacterial, etc. So as soon as the pathogen comes in contact with the receptors, the APC cells stimulate T helper using chemicals like cytokines, monokines, and interleukins. If the pathogen is exogenous, our T helper secrete lymphokines and stimulate our B cells. So firstly, B memory will form a memory and go to secondary lymphoid organs for any next interaction with the same pathogen, while the other B plasma cells will do the real work by killing the pathogen using the antibodies. The B memory forms a memory so that if there is a second interaction with the same pathogen, the lag phase, the APC contacting T helper cells will not happen, and our immune system will directly and quickly respond to the pathogen. If the pathogen is endogenous, then the T helper will stimulate T killer cells, which will attack the pathogen by using chemicals like lymphotoxins and perforin. And finally, it will finish off the job by secreting lymphokines to attract WBC cells for phagocytosis of the pathogen. Now, while all this has happened, the T suppressor cell finally give negative stimulus to all of these lymphoid cells, while T memory also makes the memory of the pathogen, endogenous pathogen, and goes to our secondary lymphoid organs. On the left side, this is our working of CMI, and on the right side, this is working of AMI. Now, if we talk about the intensity of our immunity, CMI is the strongest and innate immunity is the weakest kind of immunity.